Video we'll be talking about muscle contraction. In order for the skeletal muscle to contract, our brain will send signals from the upper motor neuron down the spinal cord, where it kind of synapses with the cell body of the lower motor neuron located at the anterior horns of the spinal cord. From here, the signal will travel through the lower motor neuron's axon until it kind of meets the axon terminal, which is next to the muscle fiber, as you can see here in this diagram. Now, at the site, where the axon terminal, let me tell you, meets the muscle fiber, we have this junction, which is called as the neuromuscular junction, right? Which houses the neurotransmitter. In this case, it's the acetylCoA. So it's this that tells the muscle when to contract. Now, before we actually continue with the actual events that happen during a contraction, let's focus on a typical muscle cell or myocyte. Myo is muscle, cyte is cell, and its functional unit, the sarcomere, right? A myocyte, let me tell you, is a long cylindrical cell with multiple nuclei, right, that are located just below the sarcolemma, which happens to be the cell membrane. Now, the sarcolemma is unique because it has these tiny T tubules, let me highlight that for you, right, that, run or project downwards from the surface towards the center of each muscle fiber, as you can see clearly in this image on your screen. The cytoplasm of, the myocyte is called sarcoplasm, right? And the myocyte actually has a specific or a special type of smooth endoplasmic reticulum, which is known as the sarcoplasmic reticulum, which happens to be the storehouse of calcium. Now, see, the sarcoplasm, let me tell you, it's filled with stacks of, long filaments. They are termed as myofibrils, and each myofibril has contractile proteins and regulatory proteins. Now, speaking about these proteins, we've got the thick myosin and thin actin. Starting off with the thick myosin, it has two parts. You've got the head and you've got the tail. The thin actin on the other end, I would say, is made up of, smaller globular proteins called G-actin, which has, these active sites, and these are the sites where, myosin kind of binds during muscle contraction. Okay. Now, these G-actins together, will form what's called an F-actin, as you see, which appears to be just a collection of these G-actins that are, kind of twisted. And let me tell you, this F-actin is in fact made up of, other regulatory proteins that include troponin and tropomyosin. Tropomyosin is more or less like a continuous filament, basically, that's shown here in orange, that's present throughout the length of our actin, okay? And the troponin, let me tell you, it's comparatively smaller, which is shown here in green, okay? And in fact, it has three subunits. You've got the T subunit, which binds to, the tropomyosin, the I subunit, which binds to the F-actin, and the C subunit that binds to calcium. Together, they form what's called the filamentous actin, F-actin, hence the name. Now, as it turns out, these thick and thin filaments, they don't, kind of run or extend throughout the length of a myocyte, but, they are arranged in these short units, which we'll call sarcomere. Now, if we were to look at a sarcomere under an electron microscope, this is what we will see. We will see, thick myosin filaments that look dark and thin actin filaments that look a little light. Okay. Now, if we were to zoom in and see the different zones, let me tell you, at the center of, the sarcomere, there is the M line, which is made up of, the myosin protein, where, the thick filaments are attached. The borders of these sarcomeres, we'll have these Z lines, where, the alpha actin proteins, where the thin filaments are attached, right? Now, there's one more thing. For every thick filament, there are, two thin filaments, one above and one below, as you can see, that they are present, and you can see overlapping also. But the region with only thin filaments is called as, the I band, which I've highlighted here. Now each sarcomere, let me tell you, has two half I bands on either ends. Okay, the region with thick filament is called the A band and it appears dark. Now most of the A band has an overlap between the thick and the thin filament, but let me tell you, there is a region towards the center which is termed as the H band and it has only thick filament, as you can see here in the diagram. Now when the muscle contracts, let me tell you, the thick filaments pull the thin filaments above and below towards the M line. Okay, even the Z line attached to the thin filament also gets pulled towards the M line, and the whole sarcomere, let me tell you, is kind of shortened. Now the A band does not change since it's the length of the thick filament, but H and I band are shortened because as the overlap increases, the region that consists of only thick and thin filament decrease, right? As you can clearly see in this diagram, as the contraction increases during maximum contraction, there is almost a complete overlap of the thick and the thin filaments, and the H and the I bands are completely gone. Okay. Now if we were to zoom in and look at how the myosin in the thick filament interacts with the actin that makes up the thin filament, we would see when the action potential kind of travels through the sarcolemma and reaches the T tubule, it kind of interacts with these interesting receptors called DHP receptors. These are interesting because they are physically connected to another receptor called the ryanodine receptors. Okay, on the sarcoplasmic reticulum. Okay, now once stimulated, the DHP receptors change shape. Okay, once they change shape, this kind of influences the ryanodine receptors to change their shape, which is a calcium channel which indirectly opens. This allows the large quantity of calcium which is stored in the sarcoplasmic reticulum just to start flowing in the sarcoplasm. These calcium ions that float over the thin filament and they start binding at the C subunit of the troponin, which is our regulatory protein that I mentioned a few slides before, which makes them change their shape. And when troponin changes the shape, it kind of moves tropomyosin basically out of its way, and that allows the F actin to kind of bind with myosin heads, right? As you can see on your screen. Now before actually myosin can bind to actin, it needs to power up. Okay. Now the part of myosin head is there is a ATP synthase, meaning it kind of cleaves the ATP molecule to ADP and phosphate, and this kind of releases energy. The energy is used by the myosin heads to get them backward into the high energy position that they are at this point, and it's bit like needing energy to kind of crank back the arm, if I can say so. Next, the myosin heads bind to the active site on the actin filament, and this is called what we say the cross bridge formation. The cross bridge formation is triggered to release the stored energy in the myosin head, kind of like firing it up, as you can see, as I've showed you here on your screen right now. And when this happens, let me tell you, the myosin heads, they launch towards the M line, pulling the thin filaments along with it, and this is called what we call a power stroke. Okay, the combination of these power strokes of all these myosin heads lead to the sliding of the thin filaments along the thick filaments, right, back and forth as you see here. Once the power stroke is done, ADP and phosphate ions leave the myosin heads, and new ATP molecule, you know, is able to bind instead, as you just saw. As soon as the ADP binds myosin heads, they kind of detach from the active site as shown earlier. The new ATP is cleaved, as you see, into ADP and IP, which kind of gets the energy to be used in getting the myosin heads, in their higher energy position and get our next power stroke. Right. Now, once the signal, sent from the lower motor neuron stops and the action potential ends, everything comes to a halt. Yeah. This is because, the calcium ion that are pumped back into the sarcoplasmic reticulum by the calcium pump that have used ATP. When the calcium, falls, the level falls, the C units of troponin is no longer bonded to calcium. So troponin returns to the original shape and allows, tropomyosin to cover up the active sites of, the actin once again. As a result, the myosin is, kind of no longer bonded to actin, and the muscle relaxes. Okay.